Hello dear students, today our topic of deliberation is structure of viruses, general structure, properties, viral envelopes and enzymes, principles of virus taxonomy. Before going through this topic, first let's have a look on its objectives. Number 1, structure of viruses, general structure. Number 2, properties, which deals with viral envelopes and enzymes. Number 3, principles of virus taxonomy. Now, let us start with the first objective, structure of viruses, which is general structure. Virus morphology has been intensely studied over the past decades because of the importance of viruses and the realization that virus structure was simple enough to be understood. Progress has come from the use of several different techniques: electron microscopy, X-ray diffraction, biochemical analysis, and immunology. Although our knowledge is incomplete due to the large number of different viruses, the general nature of virus structure is becoming clear. Virion, an entire virus particle consisting of an outer protein shell called a capsid and an inner core of nucleic acid, which is either ribonucleic acid or deoxyribonucleic acid, RNA or DNA. Virion size. Virions range in size from about 10 to 300 or 400 nanometer in diameter. The smallest viruses are a little larger than ribosomes, whereas the pox viruses like Vaccinia are about the same size as the smallest bacteria and can be seen in the light microscope. Most viruses, however, are too small to be visible in the light microscope and must be viewed with the scanning and transmission electron microscopes. General structural properties. All virions, even if they possess other constituents, are constructed around a nucleocapsid core. The nucleocapsid is composed of a nucleic acid, either DNA or RNA, held within a protein coat called the capsid, which protects viral genetic material and aids in its transfer between host cells. There are 4 general morphological types of capsids and virion structure. Number 1, some capsids are icosahedral in shape. An icosahedron is a regular polyhedron with 20 equilateral triangular faces and 12 vertices. These capsids appear spherical when viewed at low power in the electron microscope. Number 2, other capsids are helical and shaped like hollow protein cylinders, which may be either rigid or flexible. Number 3, many viruses have an envelope, an outer membranous layer surrounding the nucleocapsid. Enveloped viruses have a roughly spherical but somewhat variable shape, even though their nucleocapsid can be either icosahedral or helical. Number 4, complex viruses have capsid symmetry, that is, neither purely icosahedral nor helical. They may possess tails and other structures or have complex multilayered walls surrounding the nucleic acid. Both helical and icosahedral capsids are large macromolecular structures constructed from many copies of one or few types of protein subunits or protomers. Probably the most important advantage of this design strategy is that the information stored in viral genetic material is used with maximum efficiency. For example, the tobacco mosaic virus capsid contains a single type of small subunit possessing 158 amino acids. Only about 474 nucleotides out of 6,000 in the virus RNA are required to code for coat protein amino acids, unless the same protein is used many times in capsid construction. A large nucleic acid such as tmPRNA cannot be enclosed in a protein coat without using much or all of the available genetic material to code for capsid proteins. If the tobacco mosaic virus capsid were composed of 6 different protomers of the same size as the tobacco mosaic virus subunit, about 2,900 of the 6,000 nucleotides would be required for its construction, and much less genetic material would be available for other purposes. Once formed and exposed to proper conditions, protomers usually interact specifically with each other and spontaneously associate to form the capsid. Because the capsid is constructed without any outside aid, the process is called self-assembly. Some more complex viruses possess genes for spatial factors that are not incorporated into the virion, but are required for its assembly. Now, let us discuss the second objective, properties, that is, viral envelopes and enzymes. Many animal viruses, some plant viruses, and at least one bacterial virus are bounded by an outer membranous layer called an envelope. Animal virus envelopes usually arise from host cell nuclei or plasma membranes. Their lipids and carbohydrates are normal host constituents. In contrast, envelope proteins are coded for by virus genes and may even project from the envelope surface as spikes or papillomers. These spikes may be involved in virus attachment to the host cell surface. Since they differ among viruses, they also can be used to identify some viruses. Because the envelope is a flexible membranous structure, enveloped viruses frequently have a somewhat variable shape and are called pleomorphic. However, the envelopes of viruses like the bullet-shaped rabies virus are firmly attached to the underlying nucleocapsid and endow the virion with a constant characteristic shape. In some viruses, the envelope is disrupted by solvents like ether to such an extent that lipid-mediated activities are blocked, or envelope proteins are denatured and rendered inactive. The virus is then said to be ether sensitive. Influenza virus is a well-studied example of an enveloped virus. Spikes project about 10 nanometer from the surface at 7 to 8 nanometer intervals. Some spikes possess the enzyme neuraminidase, which may aid the virus in penetrating mucous layers of the respiratory epithelium to reach host cells. Other spikes have hemagglutinin proteins because they can bind the virions to the red blood cell membranes and cause hemagglutination. Hemagglutinins participate in virion attachment to host cells. Proteins like the spike proteins that are exposed on the outer envelope surface are generally glycoproteins, that is, the proteins have carbohydrate attached to them. A non-glycosylated protein, the M or matrix protein, is found on the inner surface of the envelope and helps to stabilize it. Although it was originally thought that virions had only structural capsid proteins and lacked enzymes, this has proven not to be the case. In some instances, enzymes are associated with the envelope or capsid. Most viral enzymes are probably located within the capsid. Many of these are involved in nucleic acid replication. For example, the influenza virus uses RNA as its genetic material and carries an RNA-dependent RNA polymerase that acts both as a replicase and as an RNA transcriptase that synthesizes messenger RNA under the direction of its RNA genome. The polymerase is associated with ribonucleoprotein. Although viruses lack true metabolism and cannot reproduce independently of living cells, they may carry one or more enzymes essential to the completion of their life cycles. Viruses with capsids of complex symmetry. Although most viruses have either icosahedral or helical capsids, many viruses do not fit into either category. The pox viruses and large bacteriophages are two important examples. The pox viruses are the largest of animal viruses and can even be seen with a phase contrast microscope or in standard preparations. They possess an exceptionally complex internal structure with an ovoid to brick shaped exterior. The double-stranded DNA is associated with proteins and contained in the nucleoid, a central structure shaped like a biconcave disc and surrounded by a membrane. Two elliptical or lateral bodies lie between the nucleoid and its outer envelope, a membrane, and a thick layer covered by an array of tubules or fibers. Some large bacteriophages are even more elaborate than the pox viruses, like T2, T4, T6 phages that infect E. coli. Their head resembles an icosahedron elongated by one or two rows of hexamers in the middle and contains the DNA genome. The tail is composed of a collar joining it to the head, a central hollow tube, a sheath surrounding the tube, and a complex base plate. The sheath is made of 144 copies of the GP18 protein arranged in 24 rings, each containing 6 copies. In T1 phage, the base plate is hexagonal and has a pin and a joint tail fiber at each corner. The tail fibers are responsible for virus attachment to the proper site on the bacterial surface. There is a considerable variation in the structure among large bacteriophages, even those infecting a single host. In contrast with the T1 phages, many coleophages have true icosahedral heads. T1, T5 and lambda phages have sheathless tails that lack a base plate and terminate in rudimentary tail fibers. Coleophages T3 and T7 have short, non-contractile tails without tail fibers. Clearly, these viruses can complete their reproductive cycles using a variety of tail structures. Complex bacterial viruses with both heads and tails are said to have binal symmetry because they possess a combination of icosahedral, the head, and helical, the tail symmetry. Now coming to the third objective of this topic, principles of virus taxonomy. Viruses are divided into different taxonomic groups based on characteristics that are related to the type of host used, virion structure and composition, mode of reproduction, and the nature of any diseases caused. Some of the important characteristics are Number 1. Nature of the host, which includes animal, plant, bacterial, insect, fungal. Number 2. Nucleic acid characteristics, which includes DNA or RNA, single or double-stranded, molecular weight, sedimentation, and number of pieces of nucleic acids, the sense of the strand in single-stranded RNA viruses. Number 3. Capsid symmetry, which includes icosahedral, helical, binal. Number 4. Presence of an envelope and ether sensitivity. Number 5. Diameter of the virion or nucleocapsid. Number 6. Number of capsomers in icosahedral viruses. Number 7. Immunologic properties. Number 8. Gene number and genomic map. Number 9. Intracellular location of viral replication. Number 10. The presence or absence of a DNA intermediate and the presence of reverse transcriptase. Number 11. Type of virus release. Number 12. Disease caused and or spatial clinical features, method of transmission. This brings us to the end of this lecture. Hope you have enjoyed and understood. Thank you and goodbye.